Right, so it's the Osmosis RPA and it's looking at some exam questions. So these are things that might be asked of you in the exam or you might be asked to explain. If you can't remember what osmosis is, the proper definition is on the slide there, and I've made a video about it that explains it in more detail. But in a nutshell, it's water going from a high concentration of water to a low concentration of water, so from lots to less. And to do that, it goes through a membrane. That membrane is called partially permeable, meaning it lets certain things in but not others, or lets certain things out but not others. So here's a quick example. There's my water, there's my sugar, there's my membrane, that dotted line. And the water is in a high concentration on the left and the right. So this will happen. High-tech animation is going to go from a high concentration to a low concentration until the concentration on each side is pretty much even. So here is the exact question, and in our case the methods, we're going to use sugar and a potato, but you could use something else. I've done it before, I've detailed it in depth, so I'm not going to go back over it, but if you want to pause the video, there's the method as a reminder. Now you could be asked certain things about it. So one of the more common questions are what are the different variables. The independent variable will be the concentration of the sugar. The dependent variable is the change in mass, that's what we're measuring each time. And the control variables are anything you have to keep the same. For example, the volume of each concentration, the size or type of potato. I could even use a water bath to make sure the temperature of each one stays the same. You might be asked, why bother to blot the potatoes? That is to dry them off each time. And that's just because you want to get rid of any excess water on the surface to make sure you're not including that in your measurements of the mass. Interesting question here, really interesting question. What about if you use the courgette or other vegetable? Would it work? Yes. What were the hazards and the risks? It's not exactly, you don't take your life in your hands doing this experiment, but you still have to use a knife, so there's a bit of a hazard there with the knife, the risk being you might cut yourself, so you might choose to cut away from your fingers, sort of like that guy's doing, but also not really. Errors, so things can go wrong, your experiment might not be perfect, perhaps your balances aren't entirely accurate, perhaps you didn't dry off the potatoes properly, and maybe some of the solution evaporated from your tube or your beaker. So again, a water bath would kind of level that out because they'd be in the same temperature. How to improve it? So this is a little bit of maths here. So even though you try and get them the same size, they're probably still not completely identical. So to make it a bit more accurate, you can try and work out the percentage change. That is, how much bigger or smaller as a percent of its original size did it get. And to do that, there's this equation, and we'll have a look at a quick example. So all you do is, here's some results for you. You take the mass at the end, which is 4.05, you minus the mass at the start, which is 6.08, and then you just divide it by the mass at the start, which I told you was 6.08. So you have a calculator, you just put these numbers in, you times it by 100 and you'll see a number like this. So it's the losing mass, so it's negative 33.388. But because I've only used 2 decimal places elsewhere in the equation, I'm going to change that to 2 decimal places here as well. So that's the equation on the board. The numbers are given to you in the exam. You just have to try and remember that, so maybe it's worth jotting it down. Right, the results. So you'll probably expect to see something like this, where the higher sugar concentrations will lose more mass and the lower sugar concentrations will lose less mass, if any. You probably will find that in just pure water it actually gains mass. It might even look a bit bigger. So why is that? So if you take a look here, we've got a potato and then kind of the blue background is the beaker, and there's a higher water concentration inside the potato than outside. So the water is going to leave the potato to the lower water concentration through that membrane, and as a result, the potato is going to lose mass because water has left it. Here, this is a slightly weaker concentration, but there's still more water inside the potato. There's still higher water concentration inside than out, so water's still going to leave the potato through the partially permeable membrane by osmosis and lose water, and as a result, lose mass. However, because the difference in water concentration on each side isn't quite as steep, it won't lose quite as much water as before. So the mass will still overall go down, but not as much as in a stronger concentration. When it's just in pure water, there's a higher water concentration in the beaker than the potato, so water is actually going to enter through that membrane by osmosis, and therefore the potato is going to gain some mass. The key thing with these questions is just look, where's the higher water concentration, and then it's going to leave that area to the other area. And if water's leaving the cell, it's going to lose mass. If water's going into the cell, it's going to gain mass. In this case, though, you can see the water concentration in to out is exactly the same. So as a result, there is no overall movement in either direction. So the potato won't gain or lose mass. And that's quite a common question when you're given a graph and it will say, what's the concentration of sugar in my potato? And it's easy. It might look scary, but it's quite easy. You just find 0, right, on your graph, the point where there's not been any gain or loss in mass, and you just go along that line sidewards until you find the line of best fit. And wherever you hit that line of best fit, that will be the concentration. So in this case, it crosses it at 0.4. So that's pretty much it. So just to check that you feel like you can describe and explain the method, you could state the different variables, you could suggest a couple of improvements, you could say where things could go a bit wrong, you could feel like you can explain the results from a table or graph, and hopefully you've not completely lost the will to live after a very boring 7 minutes. Good luck, see you later on.